Cells are complex, very complex. They are made up of thousands of distinct chemical species, each interacting in a vast network space. This complexity is what makes cells so powerful and allows them to be used as micro-machines in the field of synthetic biology. However, this complexity also poses a problem, both for scientists who try to understand how cells work and for engineers who try to use cells in a range of applications. They simply can't predict how changing one component of the system will affect the cell as a whole. The Soft Microsystems lab, led by Dr. Yuval Elani at Imperial College London, tackled this problem by deconstructing a cell into its constituent molecules and reassembling artificial cells from the bottom up. These can now be engineered with precision to internally perform chemical reactions, externally sense and respond to their environments, and communicate with real biological cells. This research is done as part of a wider effort together with other academics in the Fabry-Cell Center. One part of the group's research involves the creation of hybrid cells composed of living and synthetic matter, where artificial and biological cells are brought together as one. Another involves linking up individual cells to form intermeshed cell networks to build structures resembling tissues. To do all this group is constantly developing new tools. These include optical tweezers that act like tractor beams, which allow cells to be manipulated in 3D using light. Another is droplet microfluidics, which allows thousands of cells to be generated one by one on an assembly line with control over their content and architecture. The group also works with synthetic genomes, nanoparticles, and artificial signaling pathways. This work can make a dramatic impact in two areas. First, these cell mimics can be used as micro-machines which can deliver drugs around the body, act as sensors alerting us about the presence of toxic molecules, and act as miniature chemical reactors that can produce chemicals in tiny volumes. Second, by building simplified models of cells, we can study biological problems in a much simplified environment. After all, if you can design a cell from scratch, you can experiment how adding this ingredient or that component affects a specific biochemical process. The ultimate aim of the Soft Microsystems group is to make artificial biology by design. They hope to construct artificial systems that recreate some of biology's defining features: communication, metabolism, movement, energy generation, and replication. For more information, visit Yuval Elani online.